Thank you very much. Well, it's a pleasure to be here, to have a chance to introduce what we are doing. And actually, I'm just working 3 floors above from here, so it's rather a short distance. My world, or my research, centers around primary immunodeficiency diseases. In other words, diseases where something is wrong with your immune system. And if you have something wrong, you'll notice it, because you'll suffer from recurrent infections, and in the worst case, in the most severe forms, you simply die. So this is the beautiful architectural structure that we're living in, and we are having, in this house and in the next house, we're having one of the largest outpatient clinics for patients with primary immunodeficiency diseases. So what I'm doing is then I work as a physician as well. That means that I'm taking care of these patients. You can see them, and it's my task to make sure, or to try to find out, what's the problem? Why do they have these infections? And of course, we center around the immune system because that's where the problem lies. And once you find it, you find the spelling mistake, you find the gene that is involved in the disease, then again, it's my task to make sure that we treat them to the best possible way. So it's around immunogenetics and immunotherapy. And the story I'm going to tell you today started about 5-6 years ago. And it started with the help of a PhD student, Stefan Barter, who just graduated and who made most of the work. And he was instrumental in doing this. And the other part of the story is the Center for Metabolic Diseases, where they have the screening for metabolic diseases for all newborns in Sweden. There are approximately 100,000 children born each year, and they are all sampled. You were all sampled. You're lying in the refrigerator at CMMS, where your blood has been stored for the past 20, 30, 40 years. That means that we have access to the samples if we want to start to look at newborns and problems that have arisen in their immune system. Now, why do we want to do this? I'm going to give you a background showing that this is the immune system, where you can see that everything and anything starts from the hematopoietic stem cells. Then these cells develop and mature into other types of cells, including then those lymphoid progenitors that give rise to B lymphocytes. Those are the lymphocytes that produce antibodies or gamma globulin. They give rise to T cells that give rise to the variety of different types of cells involved in cellular immunity. It also gives rise to the natural killer cells. And of course, you have the myeloid cells and you have the erythroid cells. Now, this is a very complicated system where there are hundreds of genes that have to be turned on and turned off at the right moment. And that means, of course, according to Murphy's law, if anything can go wrong, it will. And ultimately, there will be mutations affecting a variety of these genes that will stop the development of cells in the B cell lineage, the T cell lineage, or the natural killer cell lineage. Now, broadly, when you look into immunodeficiencies, you can see that there are T cell deficiencies, which are the most severe ones, where cellular immunity is lost and where the patients come down with life-threatening infections, primarily viral infections, but also intracellular bacteria, parasites, and fungal infections. Now, there is a need for early diagnosis and therapy in those that are born with a T cell defect, because they will be born just like any other normal child, rosy, small, happy family, getting their first child or getting whatever, and they don't realize that somewhere hidden is a defect that months later will make this child severely sick and ultimately die if you're not taking care of it. There are also other types of defects, those that affect the B cell system. Those that lack antibodies, they get sick a little bit later. They are born, we're all born with antibodies from your mother, because these antibodies are being transferred throughout the pregnancy, through the placenta, into the fetus. And even if you lack the ability to make antibodies, you're born with normal adult levels of antibodies. But those will be catabolized, and within 3, 4, 5, 6 months, your maternal antibodies will be gone, and all of a sudden you will start to have bacterial infections. Sinusitis, pneumonias, and other types of bacterial infections. There's also a combination of these diseases where you can lack both T cells and B cells, and these are then referred to as combined immunodeficiencies. So you have those who are severe combined immunodeficiencies, and that is the word SCID. Anyone heard about the bubble boy? The boy that was born with a SCID some 30 years ago, and who didn't have a proper bone marrow donor, who lived in a bubble until one decided it's time to let him out, after which, of course, he died from infections. So what you have to do is to find these children early and to treat them either with gamma globulin if it's a B cell defect, and otherwise you treat them with a stem cell transplantation. And that's their only chance of survival. If you don't do that, they will ultimately die within a year or two from their severe infections. Now this is the key to the problem, because when the children are born, they look perfectly normal. But you want to find them and diagnose them as early as possible, because after a few months, when they start to accumulate their infections, things get much more complicated. And they have to be cleaned up, and their infections cleaned up before you can bone marrow transplant them, which means that if you transplant them early, they will survive. If you transplant them later and later, they have a lesser chance of survival. The one that started looking into this problem was Jennifer Puck at UCSF, who wanted to design an assay whereby you can use samples from newborns, existing samples from newborns, because, as I said, you've all been screened for metabolic diseases, but to use this very same blood sample to look for severe forms of immunodeficiency. And she invented an assay which was called TREX. And that is referred to as T-cell receptor excision circles, and is primarily the fact that when T cells mature, when you're born, you have naive T cells. They don't really know what they're going to do in life, but they have to acquire a specificity. They learn how to react against a given antigen, a given epitope, a given virus, if you want. And doing that, they have to cut and paste in their genome. And that is still what you're doing, is sitting here, you're cutting and pasting and doing a remarkable molecular biology work in your lymphocytes, because what you have to do is to take one of the gene's segments, the V genes, the J's, the whatever, and you have to put them together into a receptor that is sitting on the T cell surface, and this one would then recognize, for instance, poliovirus. This is being generated without you ever having seen poliovirus, because this is a random process and you generate a repertoire that basically covers everything. Now in doing this, cutting and pasting, there's a small piece that will be cut out. It's not being used. It's being circularized, and then this remains in the cell for some time, and when the cell divides, it expresses its receptor, its specificity, but this particular one, this T-cell receptor excision circle, what was cut out, remains as a single copy. And that means that if you look at that, it's a measure of newly formed T lymphocytes. And that means that when you're generating your immune system up to here as a newborn, you make a lot of new T cells because you need that. And as times go by, you need less and less new cells. And when you get to my age, well, life is almost over, and you don't really need a lot of new T cells. I can live with my T cells. They're going to be around for another 30, 40 years, which I may not be, but nevertheless, they're there, and they will protect me against disease. So this is a very nice system, which means that if you have no TREX, then you have no new T cells. And if you have no new T cells, you have a severe form of combined immunodeficiency. And that can be looked at by extracting DNA from the very same blood spot that is now taken from every child. This was first implemented in real life by Jack Roots, who started in Wisconsin in 2008, where they started to screen all newborn children for TREX, that is T-cell lymphopenia. Now, they found, within the first 10,000, they found the first SCID baby. And the SCID baby was transplanted, it was brought to Capitol Hill, to the politicians, and show this child would have died. Now it's been bone marrow transplanted, survives happily, and the Congress said, yes! And so there was a suggestion to implement neonatal screening using this particular assay in all newborns in the US. And today, about 2/3 of all newborns in the US, some 4.3 million per year, are being screened for TREX, that is T-cell lymphopenia. And they just published an update on the first 3 million children, where they showed that, okay, 1 out of 58,000 actually had a SCID and could be saved by this particular method, because 92% of the children survived after bone marrow or stem cell therapy. And that means that instead of dying from their disease, most of them actually did survive. They didn't miss any SCID patients, and they found a number of other less severe T-cell problems. Taiwan has been doing the same thing. They have been screening for TREX, and they found 5 children with SCID, 1 in 20,000. Again, no SCID patient missed with this assay, and some did Georges. So this is great. This works, and this could be implemented already now. But the thing is that you would only look at one side of the coin. You would only look at those deficiencies that are related to cellular immunity, that is to T cells. What we set out to do was to look at the other side, which is to look at B cell deficiencies, that is those children who have an antibody deficiency, and you can do the same thing as you do with TREX, because B cells have to rearrange their receptors too. They make immunoglobulin, but they have to have the specificity, so they have to make the receptor. Now this is referred to as CREX, kappa receptor, the light chain of immunoglobulin, excision circles, and together with a Japanese group we published that in 2011. So it's the same story. What happens is that they cut and paste, and the rest that is not being used to make this receptor, that is the thing in between that you're not using, is circularized, and this is what you can PCR amplify, and then you can see how many newly formed B cells are there. And this is also on this plot. You can see the number of B cells this way, the number of CREX. So it's sort of a linear relationship where you can also then see the number of B cells, which means that if you don't find CREX, there are no B cells. And if you don't have B cells, you have a B cell deficiency or a combined immunodeficiency. So we put it together in a test for TREX, CREX, and a marker for DNA, beta-actin, published in Blood in 2012. And we started looking at, does this assay really work? Now, mind you, this is the first assay that actually measures both T and B cell deficiencies. So we looked at retrospective samples, and we took 2,500 anonymized samples from the bank at the Metabolic Center, and you see this gray cloud. Those are the 2,500, and you can see this is the number of newly formed T cells, that is the number of newly formed B cells. And you can see they all cluster in a nice cloud. Now what happens if you start looking at patients with various forms of the deficiencies? Well, you can see that these, the green ones, that's the most severe form of B cell deficiency, is referred to as a disease called XLA, X-linked A-gamma-globulinemia, a disease where we cloned the gene some 15-20 years ago. So they're easily seen and easily picked out because they have normal numbers of T cells but no B cells. These ones should be treated with gamma globulin within 2, 3, 4 months of birth. We could also see what we set out to look for. These are retrospective samples of the children in Sweden that had been born with SCID and diagnosed with SCID for the past 20 years. And we could just go back and pick those samples, and you can see that they're all very low in T cells, which is what we wanted to see. And some of them are low in T and B cells. And by that you can conclude a little bit about what is the genetic background to this disease. And then you can see that, okay, they are RAG1s, and these up there, they're interleukin-2 receptor gammas, and they're JAK3s, and other things. So this is a very nice way of finding and also to diagnose the children. Surprisingly, we also found that you can look at ataxia telangiectasia, DOCK8, and a few other diseases, and pick them up by this very simple assay. Right. So the scene is set, or the stage is set, but we can also find that if we increase this cloud and we look at another 2,500, you can see that these are anonymized samples just taken consecutively as they come in for the screening. And you can see that, okay, most of them are in the grey cloud, but there are some that suggest that these children would be immunodeficient. Now, if you bear in mind the figure from the US, 1 in 50,000 is a SCID, you don't expect on 3,000 to 6 children who are deficient. So what are they? Well, these are the sort of additionally positive, I wouldn't have called it false positive because they are positive, they're born too early. You can see that if we take children that are born at different gestational weeks, you can see that some of them look immature or deficient, and that's what they are. So we simply wait until gestational week 40 and they normalize. We can look at a disease called DiGeorge or Catch-22, and again you can see that some of these children actually do have low numbers of T cells, normal B cells, and they're also infectious prone, so you need to find them. We have those with the trisomy 21, or as you know, Down's syndrome, and if you look at those, well, they are infectious prone, but not all that much. And that shows also here where you can see that the number of T and B cells as compared to the grey cloud is slightly shifted towards immunodeficiency. Now these are easily recognized, and that is also what was published from the US in August this year, where they found these additional positive samples being due to prematurity, DiGeorge, trisomy 21, ataxia telangiectasia, and a few other diseases. So we also find additional samples in patients with Wiskott-Aldrich syndrome, forget about that. We find them in adenosine deaminase deficiency, you can forget about that. And then we come to what is the true story then in Sweden. Well, we have this beautiful assay which works very nicely, and in Sweden we have been running the metabolic assays on newborns since the mid-60s. And the first one was phenylketonuria, where all children were screened from 64 and onwards for this particular disease. And the reason for screening, this is a metabolic disease, and the reason for screening is that if you can avoid food with phenylaniline during the first years of life or the first decades of life, you can develop normally. Otherwise, your brain will be damaged by metabolites that you cannot handle. Now, there are also a handful of other diseases that are being screened for, and most of these samples are then being stored. And this is a fantastic bank for doing research, where you can go back to samples that have been stored since the 70s and pick out whatever you want. And roughly half of Sweden's population is currently lying in the refrigerators at this laboratory. So what happens is that when you're born, within 48 to 72 hours, there's a blood sample taken. From you all, it was taken by a heel stick, and nowadays one takes a scalp stick to take the blood, and you get 4 or 5 drops of blood on a filter paper. And that is then sent in to the Center for Metabolic Diseases, and nowadays they look for 24 different diseases where they can see with tandem mass spectrometry and other methods if there is a problem in this particular field. Now, we started out then looking at prospective samples, running samples using the new test that we had developed to look for children with immunodeficiency before they developed symptoms of immunodeficiency. So back in 2010, we started with anonymized samples. We continued to do that 2011, 2012 to test the method. And in 2013, we started to do a prospective study for real, an open study. And that means that we started in November 15 last year, and we have been screening to date then roughly 30,000, because what we're doing is a pilot study in the Stockholm region. Now, we're doing a pilot study because we want to see if we can use this for screening and find patients with immunodeficiency. We're going to continue next year and the year after, and then the question is, if this works out, if we can save some babies, then this is probably going to be implemented on a national basis. Then you can say that, isn't this going to cost a lot of money to start screening children for immunodeficiencies? Well, it will. Annually, the cost for running screening for Sweden with 100,000 newborns will cost as much as building 1.6 meters of the new ring road around Stockholm. And then the question is what you want to do with your money. So we hope it will be implemented. Now the test is the first DNA-based newborn screening test in Sweden. It is the first T and B cell test for immunodeficiency in the world. And so far the results, this is a prospective study, and so far with the 30,000 we have found a few that were positive. Now most of them out of the 30,578, 30,573 were perfectly normal, which is fine because what we want to find are those with a severe form of primary immunodeficiency. So about 3 weeks ago we found the first SCID patient in the screening, and this child has no family history of primary immunodeficiency, so you wouldn't expect it. So this patient would be having his infections, and if he has a smart enough clinician, he will ultimately come to our attention as being a severe combined immunodeficiency. There was no cousin marriage. You know that in some countries where there's a lot of cousin marriage, like in Arabic countries, there will be an increased frequency of SCID because, of course, you raise the risk for inheriting the same faulty gene from both parents. It was a T negative, no T cells, no B cells, but having NK cells form of SCID. So we can sort of try to, even now, figure out what could be the genetic basis, and we're limited to maybe a handful of diseases. And of course, genetic testing is currently ongoing and will be ready within a week or two. The child is currently here, has been transferred to the bone marrow transplantation center, and he will be transplanted within very short. Now this is a life-saving operation. Without this child would have died. So what do we do with the others? We do find additional patients that are screened as being deficient. Well, they go to the doctor, and we have a dedicated doctor here at the pediatric ward who is a specialist in immunology and a specialist in looking at these diseases. He or she decides in their field what they want to do, if they want to do clinical chemistry, fax sorting to look at what are the cells there, and then we are the ones doing the genetic testing. Because, of course, you want to find out what's the background, because otherwise you cannot give genetic counseling. You have to be able to tell the parents that you have this, you have that, and that is the reason why your child is going to be sick. And you have to do that because you have to these parents are looking at a perfectly healthy child when it's born, and you have to explain why do we have to do a bone marrow transplant to save this child. So what we're doing is that we look at the screening, we screen for all the known primary immunodeficiency genes, but we don't know the whole truth, which means that we also have to look for new genes by doing exome sequencing, and in fact we are now turning into doing whole genome sequencing. And that was also advocated in one of the latest issues of Nature, where they said that the future lies within whole genome sequencing, in other words, to sequence every single base in your genome. Now this also raises some ethical concerns, because if we are to do whole genome sequencing in you or anybody else, do you want to know the truth? Do you really want to know what's in your genes, and that maybe you'll be carrying a gene that will ultimately turn you into a vegetable when you're 40? Or do you want to know that you're going to have an early form of Alzheimer? And do you really want to know from this little newborn baby, do you want to know that he or she is going to be severely sick within 10, 15, 20, 30 years? And this is one of the ethical concerns that we have to come to grips with, because the novel forms of genetic testing may tell you things that you wouldn't like to know. When I talk to my patients or to the parents, I do ask if they really want to know. And it's different. Some want to know. I did my exome. I know what I have. I know what I'm facing. But the question is, it's going to be there forever. So who is going to want to look at those data? Will the insurance companies look at it? Will others look at it? Or are we going to restrict the use of the genetic data that we're now starting to generate? Because my feeling is that within 10, 15 years, all newborns will be sequenced. Because there are a number of diseases that you can prevent, but the question is what you do with diseases that cannot be prevented. And that is also the crux when it comes to the next kind of test that we're going to do on a genetic basis using DNA for screening, and that is cystic fibrosis. And do you really want to know if you're a carrier of cystic fibrosis? Maybe not. I actually start to get requests from relatives to patients with primary immunodeficiency where they know that they have one faulty gene and I want to test my new boyfriend. Does he carry something that would make our children develop some disease? So there's a whole new world out there, and I think we better start thinking about these things before we get too much further. We are not the only ones doing screening. US started, Taiwan started, but we are the first ones to do the combined TREC and CREC, that is to look for T and B cells. Germany is following suit, England, France, and we are helping Turkey, Spain, Iran, Saudi Arabia, Oman, and a number of other countries to get started on the TREC screening. The latest edition yesterday was New Zealand. So this is the test that works. It's fine. You find the patients that you want. You can treat them the way you want with stem cell transplantation and gamma globulin, but it's not the end of the world. There are still some diseases that we do not pick up by the TREC screening, like those with phagocytic disorders and complement defects. And before I retire, 3 years from now, we're going to have assays for phagocytic disorders and complement defects on the newborns as well. And then you have the whole package of diseases. Now these are less severe, but nevertheless they cause a considerable amount of morbidity and in some cases even mortality. Finally, the acknowledgements. There are a number of clinicians who have been referring patients to us in Sweden and outside. We had some help getting started with the TREC assay, and of course things cost money, and we've been funded by the Karolinska, the County Council, VR, Jeffrey Modell Foundation, and a few other sources to get this working. So with that, I want to conclude and thank you for listening, and share with me the realization that with a single PhD student, starting from scratch, we're actually changing the world. We are making completely new things. We're starting a new screening program in Sweden. It's spreading throughout the world, and within a few years, this will be standard in a number of countries. So I'd just like to end with, think big, and the sky is the limit. You can do anything, and anything you can think of, you can do today. Thank you.